I am a dragonfly, a member of the order Odonata, suborder Anisoptera, and I have patrolled the air above ponds, rivers, and marshes for more than 300 million years. My ancestors flew alongside the earliest amphibians and dominated the skies long before birds or bats evolved. Scientists recognize more than 5,000 species of dragonflies worldwide, and each species carries subtle variations in wing pattern, body color, flight style, and habitat preference. But all of us share a fundamental design that has proven so successful that evolution has refined it rather than replaced it. I am distinct from my close relatives, the damselflies, which belong to the suborder Zygoptera. Damselflies are often smaller, more delicate, and when they rest, they typically hold their wings together above their bodies or slightly apart, while I hold mine outstretched horizontally, perpendicular to my thorax. My hindwings are broader at the base than my forewings, giving me greater lift and power, whereas damselfly wings are similar in shape. My eyes are large and meet, or nearly meet at the top of my head, granting me nearly panoramic vision, while damselfly eyes are smaller and set wide apart on the sides of the head. These differences reflect our separate evolutionary paths within Odonata, and they influence our hunting strategies, flight capabilities, and ecological niches. My body is divided into three main sections: head, thorax, and abdomen. My head is dominated by my compound eyes, each composed of up to 30,000 individual light-detecting units called ommatidia. This gives me one of the most acute visual systems in the insect world. I can detect motion, color, polarized light, and ultraviolet wavelengths, and I can track multiple moving targets simultaneously. My brain devotes a large portion of its processing power to vision, and specialized neurons filter and predict the trajectory of prey, allowing me to intercept flying insects with extraordinary precision. Between my eyes, three simple eyes called ocelli sit atop my head, helping me monitor light levels and maintain flight orientation. My antennae are short and bristle-like, reduced to mere stubs, because I rely almost entirely on vision rather than scent or touch to navigate my world. My mouthparts are adapted for chewing. I have powerful mandibles that can slice through the tough exoskeletons of beetles, moths, mosquitoes, and midges. My labium, a modified lower lip, is not extensible like that of a damselfly larva, but as an adult I use my legs to capture prey in flight, forming a basket with my spiny forelegs and midlegs, snatching insects out of the air and bringing them to my mouth while I continue to fly. My thorax is a powerhouse of muscle. It is box-like and tilted so that my legs project forward and my wings attach dorsally. I have 4 wings, each supported by a complex network of veins that provide strength and flexibility. The leading edge of each wing has a thickened dark spot called the pterostigma, which is thought to reduce unwanted vibrations and improve aerodynamic efficiency. My flight muscles are among the most powerful in the insect world, and they allow me to hover, fly backward, change direction instantaneously, and reach speeds exceeding 50 kilometers per hour in some species. I can glide for short distances, conserving energy, and I can accelerate rapidly to pursue prey or evade birds and other predators. My abdomen is long, slender, and segmented, often brightly colored with patterns of blue, green, red, yellow, black, or metallic hues. Coloration varies by species, sex, age, and even temperature. Males are often more vividly colored than females, and color can serve as a signal in courtship and territorial disputes. My abdomen houses much of my digestive tract, reproductive organs, and respiratory system. I breathe through a series of small openings called spiracles that run along the sides of my thorax and abdomen, and these connect to a network of tubes called tracheae that deliver oxygen directly to my tissues. I do not have lungs, and I do not rely on blood to transport oxygen. This system is efficient for an animal of my size and metabolic rate, but it also means I am sensitive to temperature. My muscles generate heat when I fly, and I must manage my body temperature carefully. On cool mornings, I may bask in the sun, orienting my body to maximize heat absorption, or I may shiver my flight muscles to generate warmth before taking off. On hot afternoons, I may adopt a posture called the obelisk position, pointing my abdomen upward toward the sun to minimize the surface area exposed to direct radiation, or I may seek shade and avoid prolonged flight. I live in a wide range of habitats across every continent except Antarctica. Some species prefer still waters such as ponds, marshes, and bogs, while others patrol the edges of fast-flowing streams and rivers. I can be found in tropical rainforests, temperate woodlands, deserts with ephemeral pools, high-altitude lakes, coastal dunes, and even urban parks. My life is tied to water because my larval stage is entirely aquatic, but as an adult I may roam far from water, hunting over meadows, fields, forest clearings, and gardens. Some species are highly migratory. Certain populations of the globe skimmer, Pantala flavescens, undertake multi-generational migrations spanning thousands of kilometers across oceans, riding wind currents between continents. Other species are sedentary, defending small territories around a pond or stream for their entire adult lives. My geographic range depends on my species, and within that range I select microhabitats based on temperature, humidity, availability of prey, presence of competitors, and suitability for egg laying. My day begins with temperature. I am ectothermic, meaning my body temperature depends on the environment. If the morning is cool, I remain inactive, perched on a reed or twig, waiting for the sun to warm the air and my muscles. I may bask with my wings spread, absorbing solar radiation. Once my thoracic muscles reach a certain temperature threshold, typically between 20 and 30 degrees Celsius depending on species, I am capable of sustained flight. I take off and my world becomes three-dimensional. I patrol a defined area, often a stretch of shoreline, a forest path, or an open glade. I fly back and forth, my head scanning constantly for movement. My vision is so acute that I can detect a mosquito from several meters away. When I spot prey, I calculate its trajectory in milliseconds and accelerate, adjusting my flight path to intercept. My legs form a basket, snatching the insect out of the air, and I curl my abdomen slightly to bring the prey to my mouth. I chew rapidly, consuming soft tissues and discarding wings and legs which flutter away. I do not land to eat. I continue to hunt on the wing, capturing and consuming dozens or even hundreds of prey items in a single day, depending on my size and the abundance of food. My hunting strategy varies by species. Some dragonflies are perchers, resting on a prominent twig or rock and making short, rapid flights to intercept passing insects before returning to the same perch. Others, including many hawker species, are flyers, patrolling continuously along a defined route. Flyers cover more ground and exploit scattered prey resources, while perchers conserve energy and rely on ambush. Both strategies are effective, and the choice depends on habitat structure, prey availability, and competitive pressure. I adjust my hunting height based on time of day and weather. Early morning and late afternoon, when the air is cool, I may hunt close to the ground or water surface, where warmth lingers and prey are concentrated. Midday, when thermal currents are strong, I may soar high, intercepting aerial plankton, migrating butterflies, or emerging mayflies. I am an opportunistic predator, and I take whatever prey I can catch, though I show preferences. Small flies, mosquitoes, midges, and gnats are staples. I also capture bees, wasps, moths, butterflies, caddisflies, mayflies, and other dragonflies and damselflies. I am capable of subduing prey nearly as large as myself, though this requires more effort and carries greater risk of injury. Throughout the day, I must balance hunting with territory defense and mate searching if I am a male, or with egg laying if I am a mated female. Males of many species are territorial. A male selects a section of shoreline, stream, or pond, often where females are likely to oviposit, and he defends it aggressively against rival males. Territorial disputes are ritualized and escalate in stages. First, a resident male may simply fly toward an intruder, displaying his bright colors and size. If the intruder does not retreat, the two may engage in a high-speed aerial chase, spiraling upward, looping and darting, each trying to outmaneuver the other. Physical contact is rare but can occur, with males grappling briefly before separating. Most disputes end without injury, with the intruder departing and the resident resuming patrol. Territory size depends on resource availability and population density. In rich habitats with abundant females and prey, territories are small and densely packed. In poor habitats, territories are larger, and males are more widely dispersed. When a female enters a male's territory, his behavior changes. He intercepts her, hovering nearby, displaying his colors and performing courtship flights. In some species, courtship is brief or absent, and the male simply attempts to grasp the female. In others, the male performs elaborate displays, flying in patterns that show off his agility and stamina. If the female is receptive and not already mated or unready to lay eggs, she allows the male to approach. The male uses claspers at the tip of his abdomen to grasp the female behind her head, a position called tandem. The pair may remain in tandem for seconds, minutes, or even hours, depending on species and circumstances. Before mating, the male must transfer sperm from his primary reproductive organs at the tip of his abdomen to a secondary set of structures called accessory genitalia, located on the underside of his second and third abdominal segments. He does this by curling his abdomen forward and depositing a sperm packet into his accessory genitalia. Once this is complete, and while still holding the female in tandem, he waits for her to curve her abdomen forward to bring her genital opening into contact with his accessory genitalia. This forms a wheel or heart-shaped copulatory position. Sperm transfer occurs, and in many species the male uses specialized structures on his accessory genitalia to remove sperm deposited by previous males before transferring his own. This process, called sperm competition, is a key feature of dragonfly reproduction and has driven the evolution of complex genital morphology. After mating, the male may release the female immediately, or he may continue to guard her while she lays eggs, remaining in tandem or hovering nearby to fend off rival males who would attempt to mate with her and displace his sperm. Females lay eggs in water or in plant tissue near or below the water surface. The method depends on species. Some females hover above the water and dip the tip of their abdomen repeatedly, releasing eggs that sink and adhere to submerged vegetation or the bottom substrate. Others insert their ovipositor, a blade-like structure at the tip of the abdomen, into the stems or leaves of aquatic plants, depositing eggs inside the tissue where they are protected from predators and desiccation. A few species submerge completely, crawling down plant stems underwater to lay eggs, relying on a thin film of air trapped against their bodies for oxygen. Egg-laying is energetically costly and exposes the female to predation, so she works quickly, often completing the task in minutes, though some females return repeatedly over hours or days to lay multiple clutches. My eggs are small, typically less than 1 millimeter in length, oval or spindle-shaped, and encased in a protective coating. Depending on species and temperature, eggs hatch in 1 to several weeks. What emerges is not a miniature adult but a larva, also called a nymph or naiad, a creature so different from me in form and lifestyle that early naturalists did not realize we were the same animal. My larval stage is entirely aquatic and carnivorous. The larva has a stout, wingless body, 6 legs and large eyes, though not as large as mine. It breathes through gills. In some species, gills are external, feathery structures at the tip of the abdomen. In others, including most of the larger dragonfly families, gills are internal, lining the walls of the rectum, and the larva pumps water in and out of its anus to oxygenate them. This rectal gill chamber also serves as a jet propulsion system. By forcefully expelling water, the larva can dart forward to escape predators or lunge at prey. The larva is an ambush predator. It sits motionless on the bottom, among leaf litter on submerged logs or clinging to aquatic vegetation, camouflaged by coloration and texture that match its surroundings. It waits for prey to come within reach. Aquatic insect larvae, tadpoles, small fish, worms, crustaceans, and even other dragonfly larvae. When prey approaches, the larva extends its labium, a hinged, claw-tipped lower mouthpart that shoots forward with explosive speed, impaling or grasping the prey and pulling it back to the mandibles. This strike takes a fraction of a second and is one of the fastest movements in the animal kingdom relative to body size. The larva consumes the prey, chewing it with powerful jaws, and then resumes its motionless vigil. The larval stage lasts from a few months to several years, depending on species, temperature, and food availability. Fast-developing species in warm climates may complete larval development in 2 to 3 months, passing through 10 to 15 molts. Slow-developing species in cold, high-latitude or high-altitude lakes may take 5 or even 7 years to mature, growing slowly in water that is ice-covered much of the year. During this time, the larva is vulnerable to a wide range of predators, including fish, birds, larger aquatic insects, frogs, newts, and other dragonfly larvae. Survival rates are low. Only a small fraction of the eggs laid by a female will survive to adulthood. When the larva is fully developed, a complex sequence of physiological and behavioral changes prepares it for metamorphosis. It stops feeding and moves toward the water's edge, selecting a suitable emergence site. A vertical stem, a reed, a rock, a dock piling, or even bare ground near the shore. It climbs out of the water, usually at night or in the early morning hours, a moment of extreme vulnerability. It clings to the substrate, and its exoskeleton splits along the back. I slowly extract myself, soft and pale, my wings crumpled and folded, my body fragile and damp. This is called emergence or eclosion. I pump hemolymph, the insect equivalent of blood, into the veins of my wings, expanding them to full size. I must hang motionless for hours while my exoskeleton hardens and my wings dry and stiffen. During this time, I cannot fly or defend myself. I am at risk from ants, spiders, birds, frogs, and even rain or strong wind, which can damage my wings irreparably. If I survive, my colors develop over the following hours or days, and I become what is called a tenural adult, recognizable as a dragonfly but still not fully hardened or pigmented. I leave the water and move into surrounding vegetation, where I spend several days or weeks feeding and maturing, avoiding water and staying out of sight. This period allows my cuticle to harden fully and my reproductive organs to mature. As I mature, my colors intensify. In many species, males develop brighter colors than females, and some males undergo dramatic color changes over their first days or weeks of adult life, shifting from dull browns and greens to vivid reds, blues, or metallic greens. These colors are produced by pigments in my cuticle and by structural coloration, microscopic layers that refract light to create iridescent hues. Color is used in species recognition, sexual selection, and territorial signaling. Males can assess the age, condition, and competitive ability of rivals based on coloration. Females may choose mates based on color and display quality, though evidence varies by species. My adult lifespan ranges from a few weeks to several months. Small species with rapid life cycles may live only 2 to 4 weeks as adults, just long enough to reproduce. Larger species and those that emerge late in the season may live several months, and a few species overwinter as adults in tropical or subtropical regions, living 6 months or more. My survival depends on avoiding predators and maintaining adequate energy reserves. I face threats from birds, spiders, robber flies, wasps, frogs, fish when I fly low over water, and even praying mantises and other large predatory insects. Birds are my most significant predators. Swallows, flycatchers, bee-eaters, hobbies, and kestrels hunt dragonflies on the wing, and shrikes capture perched individuals. I rely on my speed, agility, and keen vision to detect and evade attacks. My large eyes give me nearly 360-degree vision, so I can see a bird approaching from almost any angle. I can accelerate rapidly, change direction in an instant, and dive toward cover. I can also drop suddenly, folding my wings and plummeting toward the ground or water surface to escape a pursuer. Some dragonfly species have bold color patterns that may startle predators or signal toxicity, though evidence for chemical defenses in dragonflies is limited. Most of my defenses are behavioral and sensory. I am also vulnerable to parasites and diseases. Gregarines, protozoans that infect the gut, are common and can reduce my energy reserves and reproductive success. Mites sometimes attach to my body, feeding on my hemolymph or tissues, and heavy infestations can impair flight or cause death. Water mites in their larval stage parasitize dragonfly larvae and sometimes cling to newly emerged adults, though they typically drop off once the adult hardens. Fungal infections can occur, especially in damp conditions, and bacteria can invade wounds or damage to my exoskeleton. These threats are largely invisible and difficult to avoid, and they contribute to mortality throughout my life. I do not have a complex social structure. Most dragonflies are solitary, interacting with others only during mating or territorial disputes. However, some species form temporary aggregations, feeding swarms, where dozens or hundreds of dragonflies gather to exploit a localized emergence of prey, such as a mayfly hatch or a swarm of midges, are well documented. These aggregations are opportunistic and dissolve once the prey resource is depleted. Migratory species sometimes travel in loose groups, and individuals may follow similar routes and stopover sites, but they do not coordinate or cooperate. There is no evidence of cooperative hunting, food sharing, or altruism among adult dragonflies. Each individual is a solitary predator, competing with others for food, mates, and territory. Communication among dragonflies is primarily visual. Males signal territory ownership and competitive ability through flight displays, body posture, and coloration. Wing flickering, rapid hovering, and specific flight patterns are used during courtship and aggression. Some species produce sounds by clapping their wings together or by rubbing body parts, but these sounds are faint, and their communicative function is not well understood. Dragonflies do not have ears in the conventional sense, but they can detect vibrations through mechanoreceptors in their legs and body, and they may be sensitive to low-frequency sounds or substrate-borne vibrations. Chemical communication is minimal. Dragonflies do not produce pheromones for long-distance attraction, though there may be contact chemical cues involved in mate recognition or acceptance. Most communication is direct and visual, occurring at close range during encounters. I can detect the Earth's magnetic field, and some species use it for navigation during migration or long-distance dispersal. The mechanism is not fully understood, but it likely involves magnetite crystals or light-sensitive chemical reactions in my nervous system. I can also detect polarized light, which is used to identify water surfaces. Water reflects light in a polarized pattern, and I use this cue to locate suitable egg-laying sites. This ability can be disrupted by human-made surfaces such as asphalt, glass, or shiny cars, which also polarize light, leading me to mistake them for water and attempt to oviposit on them, a behavior that wastes energy and eggs and can be fatal. My ecological role is significant. As a predator, I regulate populations of small flying insects, many of which are pests or vectors of disease. A single adult dragonfly can consume hundreds of mosquitoes, midges, and flies per day, and a healthy population of dragonflies can suppress outbreaks of these insects in local areas. As larvae, we are important predators in aquatic ecosystems, controlling populations of mosquito larvae, midges, and other invertebrates. We are also prey, and we transfer energy from aquatic to terrestrial ecosystems. Birds, fish, amphibians, and other predators depend on us as a food source, and our abundance can influence their populations. We are indicators of water quality and habitat health. Because our larvae require clean, oxygen-rich water and cannot tolerate heavy pollution or sedimentation, the presence of diverse dragonfly species suggests a healthy aquatic ecosystem. Conversely, the absence or decline of dragonflies can signal degradation. My relationship with humans is complex. I do not sting or bite defensively, though a large dragonfly can pinch with its mandibles if handled carelessly, causing minor discomfort but no lasting harm. I pose no threat to people, livestock, or crops. In many cultures, dragonflies are symbols of transformation, agility, and freedom, and they appear in art, folklore, and literature. In some regions, larvae are harvested and eaten, providing protein, though this is uncommon. Dragonflies are popular subjects for wildlife photographers, naturalists, and citizen scientists, and watching us hunt and patrol is a common recreational activity in parks, nature reserves, and gardens. However, human activities threaten many dragonfly populations. Habitat loss is the primary concern. Wetland drainage, dam construction, channelization of streams, urban sprawl, and agricultural expansion destroy or degrade the ponds, marshes, and rivers we depend on. Pollution from agricultural runoff, sewage, industrial discharge, and stormwater introduces excess nutrients, heavy metals, pesticides, and other contaminants into aquatic systems, harming larvae and reducing prey availability. Climate change alters temperature regimes, precipitation patterns, and the timing of seasonal events, potentially disrupting our life cycles and range distributions. Invasive species, particularly predatory fish introduced for sport fishing, can decimate larval populations. Some dragonfly species have small, specialized ranges and are highly vulnerable to local extinctions. Others are adaptable and tolerant, thriving in human-modified landscapes such as farm ponds, golf course water features, and urban parks. Conservation efforts for dragonflies focus on habitat protection and restoration, preserving wetlands, maintaining natural stream flow patterns, reducing pollution, and creating buffer zones around aquatic habitats all benefit dragonfly populations. Restoration projects that re-establish native vegetation, remove invasive species, and improve water quality can allow dragonflies to recolonize degraded sites. citizen science programs that monitor dragonfly diversity and distribution provide valuable data for conservation planning, and they raise public awareness of the importance of freshwater ecosystems. There are misconceptions about dragonflies that persist despite scientific evidence. Some people believe dragonflies can sting, confusing them with wasps or hornets, but I have no stinger. Others call dragonflies mosquito hawks or skeeter hawks, a folk name reflecting my role as a mosquito predator. But I do not specifically target mosquitoes above all other prey. I am a generalist. Some believe dragonflies are harmful or dirty, perhaps because of their association with water and insects, but I am neither. I'm a clean, efficient predator, and I contribute to pest control. In some cultures, dragonflies are feared as omens or associated with snakes, devils, or danger, superstitions with no basis in biology. Educating people about my true nature, behavior, and ecological role helps dispel these myths and fosters appreciation and support for conservation. A typical day in my life once I am a mature adult follows a predictable pattern shaped by temperature, light, and reproductive status. I wake with the sun, perched on a twig or leaf where I spent the night. If the air is cool, I remain still, waiting for warmth. As the sun rises and the temperature climbs, I begin to warm. I may bask, adjusting my position to catch the light. My thoracic muscles reach operating temperature and I take off. I fly to my hunting area, which may be the same territory I defended yesterday, or a productive patch of meadow or forest edge if I am not territorial. I begin to hunt, scanning constantly, intercepting prey, chewing, swallowing, and hunting again. My eyes process thousands of images per second. My brain filters out irrelevant motion and focuses on the trajectory of potential prey. I am tireless in my pursuit. If I am a male and I hold a territory, I patrol its boundaries, chasing away intruders, watching for females. If I am a female and I have mated, I spend more time hunting to build energy reserves, and I search for suitable oviposition sites. If I am unmated, I may avoid male territories or move through them quickly, evading males until I am ready to mate. Midday, the sun is high and the air is warm. I may continue hunting at altitude, or I may retreat to shade to avoid overheating. I rest on a branch, my wings outstretched, my body still, but my eyes never stop scanning. I am alert to danger and opportunity. A passing bird sends me darting into cover. A cluster of midges rising from the grass draws me back into the air. Afternoon, the light shifts and the temperature begins to drop. Prey activity peaks again. I hunt intensively, capturing as much food as I can before dusk. If I am guarding a mate, I hover near her as she oviposits, ready to fend off rivals. If I am a female completing egg-laying, I work quickly, aware of the risk. As the sun sets, I find a roosting site. I land on a twig or stem in dense vegetation, away from open areas where nocturnal predators such as bats or owls might find me. I fold my wings slightly, lower my body temperature, and enter a state of rest. I do not sleep in the way mammals or birds do, but I am inactive, conserving energy until morning. This cycle repeats, day after day, until I have mated and reproduced, or until predation, injury, or exhaustion ends my life. From a family perspective, if I can speak of family in a species where parental care is absent or minimal, my story begins with my mother's choice of egg-laying site. She selected a location based on water quality, presence of aquatic vegetation, absence of fish or other large predators, and safety from disturbance. Her decision influenced my survival as a larva. Once she deposited her eggs, she departed. She provided no further care. My siblings and I hatched alone, tiny and vulnerable, each of us a lone hunter from the moment we entered the water. We were competitors as much as kin. If we encountered one another, we might ignore each other if prey was abundant, or we might attack, the larger cannibalizing the smaller. There is no sibling cooperation, no family bond. We disperse through the pond or stream, each claiming a microhabitat, each hunting and growing independently. As larvae, we molted repeatedly, shedding our exoskeletons as we grew. Each molt was a brief period of vulnerability, during which we could not hunt or flee effectively. We timed our molts carefully, often at night or in sheltered locations. Between molts, we grew steadily, our bodies expanding to fill the new, larger exoskeleton. We adjusted our diet as we grew, taking larger prey. Small early instar larvae fed on zooplankton, mosquito larvae, and tiny worms. Larger late instar larvae hunted tadpoles, small fish, and other dragonfly larvae. We were solitary, patient, and ruthless. Some of my siblings were eaten by fish, wading birds, or larger dragonfly larvae. Others succumbed to disease, failed to molt successfully, or were stranded when temporary pools dried. A few of us survived to emergence. We climbed out of the water, transformed, and dispersed. From that moment, we had no connection to one another. We might encounter each other as adults, male to male in territorial disputes, male to female in mating, but we would not recognize each other as siblings. Each of us is an individual, shaped by the environment and by chance, carrying forward the genetic legacy of our parents into a new generation. Dragonfly mating systems vary widely. Some species are monogamous for a single reproductive event, with a male and female pairing, mating, and the male guarding the female through egg-laying before separating and potentially mating with others. Other species are polygamous, with males mating with multiple females and females mating with multiple males over their lifetimes. Polyandry, where females mate with multiple males, is common and has driven the evolution of sperm competition mechanisms. Males that mate with a previously mated female must contend with sperm already stored in her reproductive tract, and many species have evolved genital structures that remove, displace, or otherwise neutralize rival sperm. Females may benefit from mating with multiple males by increasing genetic diversity of their offspring, replenishing sperm supplies if initial matings were insufficient, or receiving nutrients or other resources from males, though direct material benefits beyond sperm are rare in dragonflies. Some species exhibit alternative mating strategies. In a few territorial species, a portion of males do not defend territories, but instead employ sneaky or satellite tactics. These males lurk at the edges of territories or in vegetation near oviposition sites, attempting to intercept and mate with females before the territorial male can respond. Sneaky males are often younger or smaller individuals that cannot compete successfully for territories. This strategy has lower average reproductive success than territory defense, but it is better than not mating at all, and in dense populations where territories are saturated, it may be the only viable option. Territorial males tolerate satellites to a degree, focusing their aggression on direct territorial rivals, but they will chase away sneaks if they detect them. Parental care in dragonflies is limited. Males guard females during or after mating in some species, hovering nearby to prevent other males from mating with her, but this is mate guarding, not parental care. Once eggs are laid, neither parent interacts with them or the resulting offspring. There is no nest building, no feeding of young, no teaching or protection. Offspring are independent from the moment of hatching. This lack of parental care is typical of insects with aquatic larval stages and is related to the separation of larval and adult habitats and the overwhelming numerical odds against any individual egg surviving to adulthood. Females compensate by laying large numbers of eggs, sometimes thousands over their lifetime, ensuring that a few survive despite high mortality. Dragonfly larvae exhibit some learning ability, though it is limited compared to vertebrates. Larvae can learn to associate certain environmental cues with danger or food, adjusting their behavior over repeated experiences. For example, a larva that has been attacked by a fish may become more wary, spending more time hidden and less time exposed. Larvae can also adjust hunting tactics based on prey type and availability, showing some behavioral plasticity. However, most larval behavior is instinctive, governed by fixed action patterns that are genetically programmed. Adults are capable of learning to some degree as well. Studies have shown that dragonflies can learn to recognize individual landmarks and use them for navigation and territory defense, and they can remember the locations of productive hunting areas or oviposition sites. They can also habituate to non-threatening stimuli, ignoring repeated harmless disturbances while remaining alert to novel or genuine threats. Navigation in migratory species is a topic of ongoing research. Some dragonfly species migrate over enormous distances, crossing oceans, deserts, and mountain ranges. The globe skimmer, for instance, undertakes a multi-generational migration that spans the Indian Ocean, moving from India to East Africa and back, with intermediate breeding events. How these tiny insects navigate such vast distances without maps or learned routes is not fully understood. They likely use multiple cues, including the position of the sun, polarized light patterns, magnetic fields, and visual landmarks. Wind direction and speed also play a major role, and migrants may exploit favorable winds to conserve energy. Young dragonflies embarking on migration have never made the journey before and cannot learn the route from experienced individuals, so navigation must be largely innate, a product of evolved sensory capabilities and decision-making rules. Seasonal patterns in dragonfly activity vary by latitude and climate. In temperate regions, dragonflies are most active in late spring, summer, and early autumn, when temperatures are warm and aquatic habitats are productive. Many species have a single generation per year, emerging in early summer, reproducing and dying before winter. Their eggs or larvae overwinter, resuming development the following spring. Some species in warm temperate or subtropical regions have multiple generations per year, with continuous breeding and overlapping cohorts. In tropical regions, dragonflies may be active year-round, with breeding tied to wet and dry seasons rather than temperature. Dry season species oviposit in temporary pools that fill with seasonal rains, and larvae develop rapidly before the pools dry. Wet season species breed in permanent waters and may have longer larval development periods. Dragonflies are sensitive to changes in day length and temperature, which trigger physiological and behavioral shifts. Shortening day length in autumn can induce reproductive diapause in some species, halting egg development or larval growth until favorable conditions return. In a few species, adults enter a state of reduced activity or migrate to sheltered sites during unfavorable weather, though true hibernation or diapause in adults is rare. Most temperate zone species die before winter, and the population persists as eggs or larvae until the following season. Water quality is critical to my survival as a larva. I require dissolved oxygen levels sufficient to support my metabolic needs, which vary with temperature and activity level. Warm water holds less oxygen than cold water, and polluted or eutrophic waters can become hypoxic, especially at night when aquatic plants stop photosynthesizing and respiration by decomposers consumes oxygen. If oxygen levels drop too low, I must move to better oxygenated areas, surface to gulp air if I am a species capable of air breathing, or risk suffocation. Larvae of some species are more tolerant of low oxygen than others, and species distributions reflect these tolerances. Clear, cold, Well-oxygenated streams support different dragonfly communities than warm, turbid, vegetated ponds. pH, salinity, and the presence of contaminants also affect my survival. Most dragonfly species thrive in fresh water with neutral to slightly acidic pH, though some species tolerate brackish conditions or alkaline waters. Heavy metals, pesticides, herbicides, and industrial pollutants can be toxic to larvae, causing mortality, developmental abnormalities, or reduced growth and reproduction. Even sublethal exposure can weaken larvae, making them more vulnerable to predators or disease, or impairing their ability to complete metamorphosis successfully. Pesticide runoff from agricultural fields is a particular concern, as many insecticides are non-selective and harm beneficial insects like dragonflies along with target pests. Dragonflies serve as bioindicators, meaning their presence, diversity, and abundance provide information about environmental conditions. Healthy dragonfly communities with many species suggest good water quality, diverse habitat structure, and low levels of pollution. Declines in dragonfly diversity or the loss of sensitive species can signal degradation and prompt further investigation and management action. Monitoring programs around the world use dragonflies as indicators in wetland assessments, restoration project evaluations, and biodiversity surveys. Human fascination with dragonflies has deep roots. Ancient cultures observed our aerial prowess and incorporated us into mythology and symbolism. In Japan, dragonflies, called tombo, are symbols of courage, strength, and victory, and they appear in samurai heraldry and classical poetry. In Native American traditions, dragonflies are associated with transformation, adaptability, and the spirit world. In European folklore, dragonflies were sometimes called devil's darning needles and were falsely believed to so shut the mouths of misbehaving children, a superstition with no factual basis. In modern times, dragonflies are celebrated in nature education, conservation campaigns, and ecotourism. Dragonfly festivals, workshops, and guided walks attract enthusiasts who come to observe, photograph, and learn about us. Scientific study of dragonflies has contributed to fields beyond entomology. Our flight mechanics have inspired aerospace engineering, with researchers studying how our wings generate lift and thrust, and how our nervous system coordinates rapid maneuvers. Micro-air vehicles and drones have been designed with dragonfly-inspired wing structures and flight control systems. Our vision has informed the development of artificial visual systems and motion detection algorithms. Studies of sperm competition in dragonflies have advanced our understanding of sexual selection and reproductive strategies. Research on dragonfly migration has revealed unexpected long-distance movements and intercontinental connectivity, expanding our knowledge of insect dispersal and population genetics. Dragonfly diversity is highest in tropical and subtropical regions, where warm temperatures, abundant rainfall, and year-round breeding opportunities support numerous species. A single tropical forest stream or pond may harbor 20 or more species, each specialized for different microhabitats, prey types, or times of day. Temperate regions support fewer species, but local diversity can still be impressive, with 10 to 15 species coexisting in a single wetland complex. Arctic and alpine habitats support only a few cold-tolerant species, which have adapted to short growing seasons and freezing winters. Species specialization leads to niche partitioning, reducing direct competition. Some dragonflies prefer large, open lakes with wave action and rocky shores. Others favor small, sheltered ponds with dense vegetation. Some hunt in full sun at midday, while others are crepuscular, active at dawn and dusk. Some species patrol high above the canopy, while others skim low over water or through understory. Larvae similarly partition resources, with some burrowing in mud, others sprawling on the bottom among leaves, and still others clambering through submerged plants. These differences in habitat use, microhabitat preference, and behavior allow multiple species to coexist without excessive competition. Competition does occur, however, and it can be intense. Territorial disputes among males of the same species are common, as described earlier, and interference competition, where the presence of one individual reduces the foraging or reproductive success of another, happens when population density is high. Exploitative competition, where individuals deplete shared resources such as prey or oviposition sites, can also occur. In experimental settings, high densities of dragonfly larvae can reduce the growth rate and survival of individuals, suggesting food or space limitation. In natural settings, competition is usually less severe because populations are regulated by predation, disease, and environmental variability before resource depletion becomes critical. Predators and parasites impose strong selective pressure on dragonfly populations, favoring individuals with superior vision, flight ability, camouflage, or behavioral wariness. Birds are the most significant predators of adult dragonflies, and avoidance of avian predation has shaped many aspects of dragonfly behavior. Dragonflies with more erratic flight patterns may be harder for birds to track. Those with better peripheral vision can detect attacks earlier and react faster. Coloration may play a role, with some evidence that bright colors make dragonflies more conspicuous, but also more memorable to predators, potentially teaching birds to avoid certain species if they are unpalatable, though evidence for this in dragonflies is limited. Most dragonflies rely on speed, agility, and vigilance rather than chemical defenses or warning coloration. Larval predation is even more intense. Fish, in particular, are voracious predators of dragonfly larvae, and the presence or absence of fish in a water body strongly influences dragonfly community composition. Fishless ponds and temporary pools support different species and often higher dragonfly densities than fish-inhabited lakes. Some dragonfly larvae are active, bold hunters that move frequently and are easily detected by fish. These species dominate fishless waters. Other larvae are cryptic, slow-moving, and well-camouflaged, allowing them to coexist with fish by avoiding detection. Birds such as herons, egrets, ducks, and kingfishers also prey on larvae, foraging in shallow water and probing vegetation. Predation risk shapes larval behavior, with larvae in fish-inhabited waters showing increased hiding, reduced activity, and longer development times compared to larvae in fishless waters. The interplay between predator avoidance and prey capture is a central theme in my larval life. I must balance the need to hunt, which requires exposure and movement, with the need to avoid being hunted, which requires concealment and stillness. This trade-off influences my choice of microhabitat, my activity level, and my growth rate. Larvae that take more risks by hunting actively may capture more prey and grow faster, but they also suffer higher predation. Larvae that are overly cautious may survive longer but grow more slowly and take longer to reach metamorphosis, exposing them to risks over an extended period. Natural selection favors individuals that balance these trade-offs effectively, and the optimal strategy depends on local predator densities, prey availability, and environmental conditions. Metamorphosis is the most critical and dangerous transition in my life cycle. The moment I climb out of the water and begin emergence, I am helpless. I cannot flee, fight, or hide. I am immobile for hours, and I am conspicuous, clinging to a vertical surface. Predation risk during emergence is high, and emergence timing, site selection, and weather conditions all affect survival. Emergence usually occurs at night or early morning when temperatures are mild, humidity is high, and visual predators such as birds are inactive. Nocturnal predators such as spiders, frogs, and owls are still a threat, but overall predation pressure is lower than during the day. Larvae emerging in dense vegetation are better hidden than those emerging on exposed stems or open ground. Rain during emergence can damage soft, newly expanded wings, and strong wind can blow the teneral adult off its perch before it has hardened. Drought or sudden drops in water level can strand larvae before they reach suitable emergence sites, causing mass mortality. Once I have successfully emerged and my wings have hardened, I face a new set of challenges. I must learn to hunt on the wing, a skill that improves with practice. Young adults are less efficient hunters than experienced ones, with lower capture rates and more wasted effort. I must also learn my habitat, identifying landmarks, productive hunting areas, territorial boundaries if I am a male, and oviposition sites if I am a female. I must assess and respond to competitors and mates, and I must avoid predators that are new to me, such as birds, which were not a significant threat during my aquatic larval stage. Dispersal from the emergence site is typical. Many dragonflies move away from water for days or weeks after emergence, traveling into forests, meadows, or uplands where competition for food is lower and predation risk may be reduced. This dispersal phase allows the population to spread, reducing local density and competition, and it facilitates gene flow between water bodies. Some individuals travel only a few hundred meters, while others disperse kilometers or even tens of kilometers. Dispersal ability varies by species, with some sedentary and others highly vagile. Dispersing individuals face risks of predation, starvation, unfavorable weather, and failure to find suitable habitat, but those that survive benefit from access to unexploited resources and new breeding opportunities. Eventually, I return to water to reproduce. How I find water is a matter of sensory cues. I use polarized light to detect reflective surfaces, and I may also respond to visual cues such as the contrast between water and land, the presence of vegetation, or the overall landscape structure. In some species, individuals return to the same pond or stream where they emerged, a behavior called philopatry. In others, individuals breed at different sites, a behavior called dispersal breeding. Philopatry may be favored when natal sites are high quality and predictable, while dispersal breeding may be favored when habitats are ephemeral or conditions are variable. Genetic studies have shown that dragonfly populations are often well connected, with substantial gene flow among water bodies, suggesting that dispersal is common and effective. Sexual selection has shaped many aspects of dragonfly morphology, coloration, and behavior. Males compete for access to females, and females choose mates based on traits that signal quality, such as territory quality, body size, coloration, and display vigor. In species where males hold territories, territory quality is a major determinant of male mating success. Territories with abundant females, good oviposition substrate, and ample food attract more females, and males compete intensely for these prime locations. Larger, stronger, more experienced males usually win these contests, and they achieve higher mating success. Smaller or younger males are relegated to poorer territories or forced to adopt alternative strategies. Female choice also plays a role. In some species, females prefer males with brighter coloration or more elaborate displays, traits that may indicate genetic quality, health, or foraging ability. Females may also prefer males that provide good guarding behavior, reducing the risk of harassment by other males during egg laying. However, female choice is constrained by male harassment and forced copulation attempts, which are common in many dragonfly species. Females cannot always mate with their preferred males and must instead accept mating with persistent or aggressive males. This sexual conflict has driven the evolution of complex mating behaviors, genital morphology, and sperm competition mechanisms. Dragonflies do not exhibit parental investment beyond the initial energy females allocate to egg production. Egg size and number represent a trade-off. Larger eggs produce larger, more competitive larvae, but females can produce fewer of them. Smaller eggs allow higher fecundity, but larvae may be more vulnerable. Different species have evolved different strategies along this continuum, depending on larval environment, predation pressure, and resource availability. Climate change poses an uncertain future for many dragonfly species. Warming temperatures may benefit some species by extending their growing seasons, expanding their ranges poleward or upward in elevation, and increasing the number of generations per year. However, warming can also harm species adapted to cold environments, drying up high-altitude or high-latitude ponds and streams, or pushing species beyond their thermal tolerance limits. Changes in precipitation patterns can alter the availability and quality of aquatic habitats, with some regions experiencing more frequent droughts and others seeing increased flooding. Droughts can eliminate breeding habitat, concentrate larvae and make them more vulnerable to predators and competition, and reduce prey availability. Floods can wash away eggs and larvae, scour away habitat structure, and increase sedimentation and pollution. Phenological shifts, changes in the timing of seasonal events, can disrupt dragonfly life cycles. If larvae develop faster due to warming and emerge earlier, they may emerge before their primary prey, such as midges and mosquitoes, are abundant, reducing survival of newly emerged adults. If temperature cues and day-length cues become mismatched, it can lead to mistimed reproduction or emergence. Some species are likely to be more adaptable than others, with generalist species that tolerate a wide range of conditions faring better than specialists with narrow requirements. Range shifts are already being documented in some dragonfly species. Southern species are expanding northward in response to warming, colonizing new areas where winters have become milder. Northern species are retreating or disappearing from the southern portions of their ranges as temperatures exceed their tolerance. Montane species that depend on cold, high-elevation streams are particularly vulnerable, as warming reduces available habitat and isolates populations on mountaintop, sky islands, with no possibility of dispersal to cooler areas. These range shifts can lead to new species interactions, novel communities, and unpredictable ecological consequences. Ocean-crossing migrations by dragonflies, such as those undertaken by the globe skimmer, are remarkable feats of endurance and navigation. These migrations involve crossing hundreds or thousands of kilometers of open ocean, far from any land or freshwater habitat. Migrants must survive for days or weeks without access to water for drinking or egg-laying, relying entirely on fat reserves and opportunistic feeding on aerial insects over the ocean. How they orient and navigate over featureless seas is not fully understood, but they likely use the position of the sun, polarized light patterns, and possibly the Earth's magnetic field. Wind plays a crucial role, and migrants time their flights to coincide with favorable wind conditions, such as seasonal monsoon winds or trade winds, that aid their travel. Multi-generational migration means that individuals completing one leg of the journey breed and die, and their offspring continue the migration, a pattern seen in monarch butterflies but rare among other insects. Genetic studies of migratory dragonfly populations reveal high levels of gene flow and low genetic differentiation among populations separated by thousands of kilometers, confirming that individuals from different continents interbreed. This genetic connectivity suggests that migratory species are resilient to local disturbances, as populations can be replenished by immigrants. But it also means that widespread threats, such as habitat loss across the entire migratory route, can affect the entire species. Non-migratory dragonflies show varying levels of genetic structure. Populations separated by geographic barriers, such as mountain ranges or expanses of unsuitable habitat, may become genetically differentiated over time, leading to local adaptation and eventually speciation. However, even relatively sedentary species show some gene flow, as occasional long-distance dispersers connect populations. Human alteration of landscapes, including habitat fragmentation and the creation of isolated ponds in agricultural or urban areas, can reduce connectivity and increase genetic differentiation, potentially reducing population resilience and adaptive potential. Restoration of dragonfly populations in degraded areas is possible and has been demonstrated in numerous projects. Restoring natural hydrology, removing invasive species, replanting native vegetation, and reducing pollution can allow dragonflies to recolonize and thrive. Constructed wetlands, created for water quality improvement or wildlife habitat, can provide breeding sites for dragonflies and other aquatic insects, especially if designed with diverse microhabitats, varied depths, and native plant communities. Even small backyard ponds can support dragonfly populations and contribute to regional biodiversity, particularly in urban and suburban areas where natural wetlands have been lost. Public engagement and education are key to dragonfly conservation. Dragonflies are charismatic and accessible, and they provide an entry point for people to learn about wetland ecology, water quality, and biodiversity. Citizen science programs that recruit volunteers to survey and monitor dragonfly populations generate valuable data and build a constituency for conservation action. Educational programs that teach people to identify dragonflies, understand their life cycles, and appreciate their ecological roles foster stewardship and support for habitat protection. As an individual dragonfly, my fate is shaped by a combination of genetic inheritance, environmental conditions, chance events, and the ecological web of interactions in which I am embedded. I am the product of millions of years of evolution, a finely tuned predator adapted to a life split between water and air. I am a link in the food web, a pollinator's competitor, a mosquito's nemesis, a bird's prey, a subject of human curiosity, and a thread in the fabric of freshwater and terrestrial ecosystems. My species persists because enough of us survive the gauntlet of egg, larva, emergence, and adulthood to reproduce and pass on our genes. I am both an individual with my own brief existence and a representative of a lineage stretching back to the Carboniferous, a lineage that has witnessed the rise and fall of dinosaurs, the shifting of continents, and the emergence of humans. My ancestors thrived long before people walked the earth, and if humans act wisely, my descendants will continue to patrol the waters and skies long into the future.